I'll do a brief intro for you, Jeff, and before you get started. Hi, everyone. Thank you again for coming to the Spatial Omics Seminar Series 5. And today we are very excited to have Dr. Jeffrey Moffitt from Boston Children's Hospital and from the Department of Microbiology in Harvard Medical School. And Dr. Jeff Murphy started doing this kind of MERFISH actually when he was a postdoc at Harvard working with Dr. Shui Zhuang, and where they co-invented MERFISH technology. And currently he has his lab at the Children's Hospital, and he's currently an assistant professor of program in cellular and molecular medicine and Department of Microbiology. And his lab has been doing mapping the neural circuits that transmit gut state to the brain. And since it's going to be an exciting talk, I would like to keep this part short for now. But I mean, he's amazing in terms of MERFISH technologies. And today we will be hearing about the applications to various different fields. And without further ado, I'd like to turn to Moffitt, Dr. Moffitt.
Ahmet, thank you so much for the introduction. And also thank you and to Rong as well for the introduction. This has been a phenomenal speaking series that you guys have put together. It's very humbling to be a part of this because it is a really illustrious group of speakers and amazing talks that you've assembled and posted online. So I'm happy to be a part of this, and I really appreciate this invitation. And I'm thankful to everybody who's tuned in today to go through this talk with me. So what I'd like to do today is to give you an overview of a technology that I co-developed as a postdoc. It's the technology multiplexed error robust fluorescence in situ hybridization. It's a mouthful, so we're going to call that MERFISH from now on. And I'm going to give you a brief introduction to how this technology works, and then I'll give you two applications, one that's published and just a hint of some unpublished stuff that we're very excited about. In a nutshell, MERFISH is a spatially resolved form of single-cell transcriptomics. It is a method that allows you to image and identify individual RNA molecules within intact samples at the scale of hundreds to thousands to now tens of thousands of different molecules. And with those spatially resolved transcriptomic measurements, define and map cell types and create tissue atlases, as well as a wide variety of additional applications. But today I'm going to focus on the tissue atlasing effort. So before I get into the details of the talk, though, I just want to disclose that I am a co-founder and an advisor and a stakeholder in a new startup company called VizGen, which aims to commercialize the MERFISH technology, though none of the products or services of VizGen were used to generate any of the data that I will present here today. So I want to start by giving you a sense of why one would want spatially resolved transcriptomic methods. And so I'll do this by sharing with you a story from when I was young. I used to like to do a lot of auto mechanics with my father because I was always fascinated with how complex machines work. And there's really nothing more complex in my mind than the car engine. And so with that in mind, you can understand why when one day I turned the key in our Ford Ranger and this horrible smoke poured out the tailpipe that I was so absolutely thrilled, because white smoke, which is what came out of our tailpipe, means that you're burning oil, which means that you've broken the head gasket. There's one and only one way to fix that, which is to pull the engine out of the car and completely disassemble it. And we did that. We pulled the engine out of my father's Ford Ranger and had it completely disassembled on the floor of our garage. And there was a sense with this engine disassembled that I truly understood how it worked because I could understand its parts. I could see the pistons, the spark plugs, the camshaft, everything. But you realize when you have to then reassemble this engine that knowing these parts is really only the first part in understanding how these complex systems work. You need to know how they fit together to give rise to the whole. And we are really living in the era of disassembly in biology. With this remarkable suite of technologies known as single-cell RNA sequencing, we can take almost any complex biological system, disassociate it or disassemble it into its individual parts, in this case cells, extract from those cells the RNAs they express, and use next-generation sequencing to provide genome-scale profiles for individual cells, which allow you to then computationally define and catalog the types and states of cells that are present in the tissue. And so now you can define their parts. You can tell what those parts are doing, and you can understand functionally aspects of their behavior based on the genes they express. But in dissociating these cells from tissues and extracting RNAs from cells, you've lost a sense of how these systems are assembled. You've lost spatial context. And that is essential to so many different aspects of biology. And I want to give you a sense of the two different types of context that you've lost. So here on the left, what you see are 9 images taken from stains of the mouse brain. This is from the Allen Brain Atlas. In each of these images, we've stained a different RNA that marks different populations of cells. And I hope you can clearly see that each of these marker genes are defining different cell populations. Single-cell RNA sequencing would have had no problem defining these populations, but would have failed to tell us that they are organized into this beautiful layered pattern that we know is essential to cortical function. But there's another aspect of spatial context as well, which is captured by this image. What you see here is a single crawling fibroblast cell. It's crawling from the lower left of the image to the upper right. And in color, what we've done is mark the internal concentration of a single RNA, the beta-actin mRNA. And what I want you to see here is that this RNA is not just uniformly distributed throughout the cytoplasm, but is strongly enriched at the leading edge of the cell, exactly where the cell needs beta-actin. And we now know that this type of intracellular organization is not the exception. In many cases, it's the rule. And it will produce a very potent form of post-transcriptional regulation for many RNAs in many biological processes. And so I think you can clearly see from these images that knowing a catalog of the types of cells that are present in a tissue and a list of the RNAs that are found within the cell is simply not going to be sufficient to understand a wide variety of biological questions. What we need are methods that capture the power of transcriptome scale measurements, but while preserving this essential spatial context. We need spatially resolved single-cell transcriptomic methods. And it was with that in mind that we set out to build MERFISH. So today I'm going to now tell you a little bit about MERFISH. What's the basic concept? How have we implemented it? How has it performed? And then to give you a taste of the type of applications that one can use MERFISH to address, I will tell you about how we've used this technology to map out the spatial organization in a portion of the mouse brain. And at the very end, I'll give you a hint of how we're now doing this in other tissues. So MERFISH is based upon a very powerful technique known as single molecule fluorescence in situ hybridization, or FISH. In that approach, you fix your sample, permeabilize the cell membrane, and then hybridize to that system a set of fluorescently labeled DNA oligonucleotide probes, complementary to different regions of the RNA of interest. This base pairing of these probes concentrates a large number of RNAs or probes to each copy of that RNA, which produces a nice bright fluorescent spot when you image the sample with an epifluorescence microscope. So this immediately tells us the number of RNAs found within each cell. You just count the number of spots and their location because these samples are fixed. And our goal with MERFISH was to take this powerful technique and massively multiplex it. This is how we do so. We assign to individual RNAs a unique binary barcode, and then we read out those barcodes with a series of single molecule FISH images. In the first image, we stain every RNA that has a 1 in the first bit of the barcode assigned to it. That'll produce a pattern of fluorescence like this. We image the sample, remove this signal, and restain the same sample. This time targeting every RNA that has a 1 in the second bit of its barcode. We get a different pattern of fluorescence. We image, remove, and repeat with 1 round of single molecule FISH imaging for every bit in our barcode. That fluorescence on-off pattern that you observe for each RNA then determines the binary barcode and thus allows you to identify that RNA. Now, this process is not error-free. Every now and again, you have an RNA that does not accumulate enough probe to be bright enough to call it as an RNA, or some background fluorescence that tricks you into thinking an RNA is present when it's not. That can be thought of as corrupting or misreading bits. And a crucial aspect of MERFISH that we introduced into this spatially resolved field was that we're going to use binary barcodes that are robust to these type of errors, that can actually allow us to detect when bits have been corrupted, and in many cases, uncorrupt those bits, effectively correcting the measurement. So let's take a look at some actual MERFISH data. That's what you see here on the left. It's a culture of osteosarcoma cells. There are 3 cells in this image. And this is after the first round of MERFISH staining and imaging. So each bright fluorescent spot is a single RNA molecule. If we zoom in on one small portion of one of these cells, we can see one of these molecules, and its presence in this image gives us a 1 in the first bit. We remove the signal, restain, and now other RNAs are fluorescent, but that RNA is no longer fluorescent, giving us a 0 in the second bit. We repeat. In the third round, this RNA is fluorescent again, gives us a 1 in the third bit. And at this point, I think you get the idea. We're going to do this 13 more times to build up a stack of 16 images, all of the same sample. And the fluorescent on-off pattern for this RNA gives us this binary barcode, which identifies it. Of course, we have a barcode like this for each of the 140 different RNAs we targeted in this measurement, allowing us to reconstruct an image like this, where each marker represents a single RNA molecule and the color is its identity. And so this is the way in which we can optically barcode and identify individual RNA molecules. Now I want to go a little bit deeper on how we've actually implemented MERFISH. So in traditional single molecule fish, you will bind fluorescently labeled probes, many tens targeting different regions of an RNA to give you binding redundancy. In MERFISH, we tile probes in the same fashion, but instead of having fluorescently labeled probes, we have these unlabeled probes that we call encoding probes. The central part of these probes is complementary to the region of the RNA that we want them to bind to. That's what we call a targeting sequence. And then flanking this are barcode regions. Our barcodes are comprised of what are known as readout sequences. These are 20 nucleotide optimized binding sites with one unique 20 nucleotide binding site for each bit in your barcode. The presence or absence of a readout sequence corresponds to a 1 or a 0 in the corresponding bit. So the specific readout sequences that are present on an encoding probe is literally the physical embodiment of the barcode. Now, the advantage here is that when we want to read out a specific bit, we can hybridize on a single fluorescently labeled readout sequence or readout probe complementary to that readout sequence. Its presence will tell us that that RNA has those readout sequences and thus has a 1 in that corresponding bit. The advantage here is that the binding of these probes is extraordinarily fast. Here you can see the occupancy of probes versus time. In minutes, they're fully occupied. So this means the process of repetitive staining can be very quick. When we're done imaging the sample, we have to get rid of this fluorescence. We do that by linking our fluorophores to our probes via disulfide bond so that a gentle reducing agent can cleave that bond and we can wash these fluorophores off of our sample in one go. This shows you the brightness of spots in exposure to this reducing agent TSEP. Again, rapid cleavage and removal. So really in just minutes, we can reset our sample for the next round of imaging. That's essential for the type of throughput that I'll show you we have. I also want to just highlight that if you have multiple colors of fluorophores, you can assign individual colors to individual bits in specific channels or specific readout rounds so that you can do multiple bits in each round. So to understand how well MERFISH performs, we made these measurements across 140 different genes across hundreds of cells, computed the average expression level for each of those RNAs determined by MERFISH, and compared this with an independent technique, in this case, bulk RNA sequencing. That's what you see here. On the left, MERFISH, on the x-axis, bulk RNA sequencing, and each marker is a single gene. So these two technologies correlate very strongly, telling us that we can measure relative abundances accurately. To determine absolute abundances, we perform these measurements and compare them against the gold standard for copy number measurements in single cells, which it turns out is conventional single molecule fish. We did those measurements for 15 different RNAs, and these are the results here. Again, you see these two technologies correlate very strongly. But there are a few things I want to highlight on this plot. First, MERFISH agrees with single molecule fish across the full dynamic range of single molecule fish. So we can measure RNA abundances that span at least 3 orders of magnitude. The agreement goes all the way down to less than a single copy per cell. So this is a very sensitive technique. And you'll notice that these markers fall along the dashed quality line, telling us that we have near 100% detection efficiency of the molecules that are present in the sample. And if you think about it, it's actually pretty remarkable because what I've told you is that we can increase the multiplexing of single molecule fish by at least 100-fold with almost no cost in the performance of that technique. So inspired by that, we've continued to push the multiplexing of this technology forward. And I want to highlight you some work that came from colleagues of mine in Xiaohui's laboratory. What you see here is a culture of, again, osteosarcoma cells. But instead of targeting 140 RNAs, this is 10,000 different RNAs. The central challenge here is RNA density. As you target more and more RNAs, the fluorescent signal from those RNAs start to overlap. And if they overlap too much, you cannot decipher the barcode. One way in which you can solve this is to couple MERFISH with expansion microscopy, a technology that allows you to physically swell your sample, diluting those RNA signals. And that was one part of how this problem was solved. Another way to solve density is to increase the number of bits in your barcode while keeping the number of rounds in which RNAs are fluorescent fixed. That number of rounds is called the Hamming weight, and you can dilute out these signals by keeping a fixed Hamming weight, but longer barcodes. That's the advantage of this binary encoding scheme. You have a lot of flexibility in how you choose your barcodes. And what you can see is that Xiaohui's lab was very nicely able to identify 10,000 different genes in single cells while preserving the excellent performance relative to bulk sequencing and the high detection efficiency. And so MERFISH is a technology that can make sensitive and accurate measurements of the vast majority of the expressed transcriptome, if that's what you need in your measurement. So with this ability to perform massively multiplex gene expression measurements, you actually open up a wide variety of very exciting biological questions to direct interrogation. I want to give you a taste of the questions that we've started to tackle with this technique. So because you capture almost 100% of the molecules that are present within a cell, you can actually now leverage the natural variation in gene expression from one cell to the next to infer properties of regulatory networks. The idea being that if a transcription factor or another regulatory element varies from one cell to another, that will propagate into correlated expression of all the genes that it co-regulates. So covariation in gene expression can allow you to back extract regulatory networks. And we've shown that this works very nicely, and you can now assign function to genes based purely on their correlation of genes of known annotation. But of course, you don't know just how many copies of an RNA are found within the cell. You also know exactly where each and every one of those copies was found. So you immediately can map the internal organization of the transcriptome, and that's yielded some very interesting insights into a range of questions. You can also infer aspects of cell state based on the genes that are expressed or the location of those RNAs. Every RNA starts its life in the nucleus and is degraded in the cytoplasm. So the ratio of nuclear to cytoplasmic RNA copies can actually tell you something about the transcriptional history as well. You don't have to measure just native RNAs. You can measure synthetic RNAs. Those synthetic RNAs can serve as barcodes. For example, you can create a pooled library of genetic perturbations where every cell carries a specific perturbation. It also expresses a unique synthetic RNA barcode. Now you can array that library on a cover slip, characterize the phenotype of those perturbations with microscopy, and then use MERFISH to go back and cell by cell determine the genotype based on the barcode that they express. And finally, you can do these measurements in intact tissue samples, discover and map cell types. And this is the application that I'm going to tell you about for the remainder of my talk. So the specific tissue that I'm going to tell you about now is a portion of the mouse hypothalamus known as the preoptic region. This is a coronal and a sagittal section of the mouse brain, and the preoptic region is in this black box and around this dashed line. Now, this is an interesting brain region because it contains circuits that are responsible for a variety of unlearned and instinctive behaviors. So how a mouse responds to homeostatic stimuli, hunger, thirst, fever, or even more complicated stimuli that trigger social behaviors, aggression or mating or parenting. And because this region of the brain contains circuits that control these unlearned behaviors, it stands to reason that these circuits are derived from transcriptionally distinct neuronal types. And so we wanted to ask a simple question. What are the type of neurons that are found within this region of the brain? How are they spatially organized? And perhaps more interesting, how are they functionally organized? How does a small number of neuronal types give rise to this wide variety of hypothalamic functions? And in many ways, MERFISH is perfectly suited for these questions. But because this application was our first time mapping out at large scale cell types in the brain, we wanted to have a reference against which we could benchmark MERFISH. So we actually did single cell RNA sequencing in this brain region. We sectioned out the preoptic region from multiple male and female mice, dissociated and captured 31,000 cells, characterized their gene expression with the Chromium platform. And that produced the data that you see here. Genes are individual rows, cells are columns, and color is the expression of those genes in those cells. We clustered these with standard graph-based Louvain methods and project them using t-SNE. That's what produces this plot here. And the color is, of course, the cluster identity. And so what we find in these data are all of the types of cells you'd expect to see in this region of the brain. And if you subcluster your inhibitory neurons in red and your excitatory neurons in yellow, you derive about 65, 67 different neuronal types in this region of the brain. So with that benchmark data set in place, we decided to now perform MERFISH of 150 different genes. And we divided up our panel in the following way. About 30 of our genes were classic markers of known cell types. So genes added to discriminate inhibitory neurons from excitatory neurons, from oligodendrocytes, and so forth. About half our panel was actually derived from our single cell data. We wanted to see if we could include markers that allow us to discover these same neurons we'd found in that data set. But we didn't want to just define neuronal types. We also wanted to learn something about their biology. So the rest of our panel was filled out with a comprehensive set of neuropeptides and neuropeptide and hormone receptors, because these are important modes of communication between neurons in this region of the brain. And this class of receptors actually is a very important group here, because receptors are a class of genes that can be very functionally relevant, even when they're expressed at very low levels, a few copies per cell. And while single-cell RNA sequencing has many strengths, one of its potential drawbacks is it often captures a modest fraction of the transcriptome, say about 10%. So it can struggle to characterize and extract information based on functional genes that are expressed at low levels, just like these receptors. By contrast, MERFISH has single molecule sensitivity, so we had hoped to do a better job of characterizing this important class. I'll show you that we do here in a few slides. So now what we did is we dissected out 2 millimeter by 2 millimeter by 600 micron blocks in the preoptic region, sectioned those into 10 micron thick sections, and characterized every fifth section. Here, what you see on the left is a MERFISH measurement from one of those sections. This is a 200 by 200 micron field of view. Each white dot is a single RNA molecule imaged anywhere within the 10 micron thickness of that slice. We can now color these molecules based on their identity as determined via MERFISH, and we can define the boundaries of these cells using automated segmentation routines. That produces the image that you see here. And I love these images because you can immediately see the transcriptional heterogeneity across even such a small region of this tissue. You have cells that are predominantly making red RNAs, green RNAs, or purple RNAs. And we're directly visualizing the unique molecular fingerprints that discriminate one type of cell from another. Now, to give you a sense of the scale with which we can do these measurements, let's zoom back to a single 2 by 2 millimeter slice. That's what you see here. And the white box is the field of view that I just showed you. Now, because we can't comprehend a color palette of 155 different colors, I'm just plotting 8 RNAs for simplicity. I want to give you a sense of the transcriptional heterogeneity we see across these large tissue areas. And so we're going to walk through the distribution of these 8 different RNAs. And I hope you can clearly see that they're not just defining cell types, but anatomical features in these tissue slices. Now, because we have a boundary of these cells and we know exactly where each RNA was imaged, we know how many RNAs are found within each cell, and we thus have an expression profile, which means that we can now analyze these data using the full statistical toolbox developed for single-cell sequencing, unsupervised clustering, and t-SNE visualization. That's what produced the image here on the left. But because MERFISH is such a high throughput technology, as opposed to having about 30,000 cells, which we had in our single-cell sequencing data set, here we have 500,000 cells. And now we can ask an essential question. MERFISH measured 150 genes. Is that enough to define the heterogeneity of cell types in this region of the brain? And what you can see from this UMAP is it absolutely is enough. We find all of the classes of cells we'd expect to find. In fact, we even have a population of cells called ependymal cells that I'll point out here in a second that we struggled to see in our sequencing data, presumably because they were lost in the dissociation. So now we've defined these cell types with 2 very different technologies. One where you dissociate cells, extract the RNAs and sequence them. Another where you would image and identify them in situ. We found both cell types in both data sets, but now we can ask, to what degree do the data sets agree with one another? And so we computed the average expression profile for each of these cell types in MERFISH and in sequencing. MERFISH is on rows here, sequencing is on columns, and we computed the pairwise Pearson correlation and coefficient between them. So the fact that we see a strong diagonal tells us that these 2 different technologies not only produce the same cell types, they produce quantitative agreement in the expression profile of those cell types, which is actually a beautiful cross-validation, not just of MERFISH, but of sequencing as well. So MERFISH can define cell types, but the advantage we have is we've imaged those cell types in their native context. So we get an atlas of this tissue essentially for free. That's what you see here. Each black box is a 2 by 2 millimeter slice taken from one portion of the preoptic region of a single mouse. And these 12 slices are all from the same mouse, arrayed anterior to posterior, left to right. Gray marks the boundaries of each of the 60,000 cells imaged in this single data set, and we can now color them based on their identity determined via MERFISH. So these are our inhibitory neurons, which are distributed throughout the brain, but depleted in structures in the top third. These are excitatory neurons, which very interestingly have a completely unique spatial distribution. These are myelinating mature oligodendrocytes, which are distributed throughout the neuron-rich regions, but heavily enriched in these regions, the top third of these slices depleted in neurons. It's very nice to see. These are marking a known structure called the anterior commissure. That's a nerve fiber tract, and it's exactly the type of structure where you expect to see myelinating oligodendrocytes enriched and neuronal soma depleted, exactly as we see. We have a precursor population of oligodendrocytes, which very interestingly has its own unique spatial pattern, telling us that there must be a spatial component to the maturation of these cells. We have microglia, astrocytes, endothelial cells all distributed throughout the brain, pericytes, which are involved in blood flow through the brain. We see these structures. And here's this population of ependymal cells that was largely lost in our single cell sequencing data. they form this beautiful single cell layer flanking a structure known as the third ventricle. These are ciliated cells, and they drive the movement of cerebral spinal fluid, which is what is found in the ventricle. This is exactly where these cells are known to be found. And of course, you can put back all these colors to reconstruct an atlas of these major cell classes in a single mouse's brain. Now we can take our inhibitory and excitatory neurons and subject them to a second round of clustering. And what we find are 70 different types of neurons in MERFISH. And again, we can do the same type of Pearson correlation analysis, comparing MERFISH neuronal types to those derived in sequencing. That's what you see here. Again, MERFISH in rows, sequencing in columns, and this strong diagonal, again, cross-validates both of these measurements. But now we can do one step better and ask, can we find a correspondence between the neuronal types we found with one technique and the neuronal types we found with another? And so we did the absolute simplest thing you could imagine. We pulled out the largest and statistically distinct correlation coefficients from that matrix, leaving the elements you see here, and the non-zero elements in this matrix are our correspondence between neuronal types identified in one data type and the other. What I want you to see is that the 80% of the neuronal types we identified with MERFISH have a corresponding type in sequencing. Again, a nice validation. But I want you to also notice that this correspondence is not always one-to-one. There are instances where one technology produces a neuronal type that agrees with multiple neuronal types from the other technology. And what we think is happening here is that one technique is better able to subdivide neuronal types that are similar than the other technique. That's unsurprising. MERFISH measured 150 genes. Sequencing had access to the whole transcriptome. But what is perhaps surprising is in the vast majority of cases, it is MERFISH that is better able to subdivide these neuronal types as compared to sequencing. And so we decided to examine why that might be in at least a few cases. So what you see here are two sets of neuronal types. These are types that we correspond between MERFISH and sequencing. These are all positive for the neuropeptide galanin. This is a dot plot showing you the expression of the genes that are listed here on the left in each of these neuronal types. And it may be a little bit difficult to see, but the color of these columns maps this correspondence. So this green column represents that little e3 and capital E8 agree between the two data sets. What I want you to notice is that there is a type of neurons here in MERFISH, I14 and I16, that agree with a single type in sequencing. This is an instance of where MERFISH has been able to subdivide neuronal types. If this were actually happening, we could look and ask, what are the genes that distinguish these two neuronal types? That's what you see plotted here. And what distinguishes them largely are these receptors, neuropeptide and hormone receptors. Very lowly expressed, but very important in defining the functionality of these neurons. Now, if this division was real, presumably there is a statistical signature of it within this single neuronal type found in sequencing. So what we did is look for that signature by looking at the correlation and expression between these genes across these cells. Here you can see the block of genes that define I14 and I16 are anti-correlated. If you look at this sequencing neuronal type, you see the same anti-correlations, even though sequencing was unable to subdivide those neurons. And so that supports this division. And I'll show you here in a slide that we have further functional support for this division. And so what I want to emphasize here is that it's not just about space, it's also about sensitivity. There is, of course, I'm sure everyone would not be surprised with this. There's real biology in these lowly expressed genes that are only a few copies per cell. Having access to those expression can give you insights into biology that you may miss with technologies that are not as sensitive. Now, of course, we can ask how these neurons are distributed throughout the brain. There's a lot we've learned in the hypothalamus in terms of basic hypothalamus structure and circuit function. I don't want to say any of that today. I just want to highlight what we observe. So again, an atlas, 12 slices arrayed anterior to posterior, left to right. Now we're marking in green, red, and blue each instance of 3 of the more abundant inhibitory neurons. You can see the patterns are bilaterally symmetric and that they change remarkably quickly over relatively short distances. In addition, they're beautifully reproduced across replicate mice. Here are 3 more neuronal types. These are excitatory, and here are 3 peptidergic neurons. What I want you to see here is that there is a remarkable spatial richness. What we find is that the overarching organizational principle in the hypothalamus is that any small region of the hypothalamus is not defined by a unique set of neuronal types, but rather the intersection of multiple neuronal type distributions. Now, there's one more thing that we can say in terms of space. So for any of you who have done single-cell RNA sequencing or any of these single-cell technologies, you know that even within a uniform population of cells of a single type, there's variation in gene expression. And that could be real biology or technical variability. But with MERFISH, we can ask if any of that variation is due to spatial location. So what you see here on the left is, again, a single slice from the mouse brain. Gray are all cells, and red are each instance of a specific neuronal type. We can now pull out those neuronal types, decompose their gene expression with principal component analysis, and replot them, now colored by the strength of their first and second principal component. What I want you to notice is that large components of the variation in gene expression within these cells is actually dictated by their location in the brain. What's exciting about this is not that we can say that this is functionally relevant, but that we can now measure this. And it could be that this type of small variation in gene expression gives rise to functional variations that are important for the function of these circuits. The fact that we can measure this means that we can now ask those questions. Okay, so we've discovered cell types, we've mapped them out. Can we say something about their function? And so what we did was a very simple experiment. We took a female mouse, we added a pup to its cage. That mouse will actually go and retrieve the pup and bring it back to its nest, even if it's not its own pup. That's what's called the instinctive parental response. And then after that very heartwarming display, we sacrifice that mouse and we perform MERFISH in its brain. And we add one gene to our panel, the gene CFOS. Now, CFOS is added not to discriminate neuronal types, but because it is what's known as an immediate early gene. These are genes that are rapidly transcribed in response to neuronal activity. So the expression of CFOS is now a proxy for neurons that were active during this parental display. Now what we can do is find the 70 type of neurons and ask how many of each of those neurons are CFOS positive in that mouse. That's what you see here. Each bar represents a different neuronal type, and the level of the bar is the CFOS positive fraction. And I still find this remarkable, but what we find is in these virgin female mice, one and only one neuronal type is CFOS, has a statistically significant enrichment in CFOS positive cells during this parental display. Now we can do the same experiment, but with a male mouse. We add a pup to its cage. It will often attack and kill that pup. It displays an aggressive response. We do MERFISH in its brain, and we find that even though this parental neuron is present, it is not CFOS positive, but rather a different set of neurons, presumably underlying this aggressive response, are now positive. And now we can do one very interesting experiment. We can take that male mouse 3 weeks after sexual experience. It will actually care for that pup just like a female. When we do MERFISH in its brain, this parental neuron is now active again. But interestingly, the aggressive neurons are also active. So the most simplistic model is that this mouse has a native aggressive response that is overwhelmed by a state-dependent switch that turns on the parental circuit. And what I want you to take from this is that we can now stimulate these mice with a wide variety of hypothalamic stimuli and now functionally annotate our neurons, which we've done here, including additional features of these parental and aggressive responses, as well as mating. And so now we can provide a link between the specific identity of these neurons and their function in these circuits. And I want to leave you with one last point. This parental neuron here and aggressive neuron here, these were the two neurons that MERFISH was able to subdivide, whereas sequencing put them into the same type. So if we had only used our sequencing data, we would have come to the conclusion that a single neuronal type drives both parental and aggressive responses. In reality, we now know that they are very similar neuronal types that are distinct in a small number of receptor genes, and yet they have these very different functional roles in the hypothalamus. So what I want you to take from this is that we created a molecularly defined atlas of the preoptic region of the hypothalamus, and that revealed to us a large number of distinct neuronal types that are largely selectively activated in different hypothalamic behaviors, which favors a model in which the large variety of specific hypothalamic functions are supported by a similarly large number of transcriptionally distinct neuronal types. We found the spatial organization of this region of the brain, and the specific intermixing we see raises some very interesting implications for the interaction between neurons in the circuits that are found in this region of the brain. And what I hope you take from this is that MERFISH is a powerful method for the construction of cellular atlases, potentially in a wide variety of tissues. And with that, I want to just end with a little bit of a preview of some preliminary data that my lab is now collecting, highlighting this ability of this technology to map other tissues. And so what you see here is a slice of the adult mouse gut in which we've characterized several hundred genes with MERFISH. And you can see the spatial distribution of these genes in this image over here. Each marker, of course, is a single gene, and its color is its identity. Here we're plotting only 8 genes again for simplicity. Once again, we can compare the average abundance determined by a MERFISH to that determined by a bulk RNA sequencing. And again, these two technologies agree very nicely, showing that we can recapitulate gene expression with MERFISH in other tissues. We can take a closer look at the distribution of RNAs within this small portion, and I want to just give you a sense of the richness of these data. So here, each gray dot represents a single RNA, and in each of these frames, blue represents each instance of the RNAs that we see here. So not only are we finding good agreement in the abundance of these RNAs, we are seeing that classic markers of specific cell types and features in the gut are found exactly where we would expect to find them. Now, one of the challenges that we face when we move from the brain to other tissues is that in many ways, the brain can be a relatively cellular sparse tissue. As you saw in those images, individual neuronal soma were largely dispersed. But in many other tissues, you actually have very cell-dense regions where you have many different types of cells all pressed closely together. And of course, in any of these image-based transcriptomic methods, one needs to be able to assign RNAs to individual cells to be able to determine the expression profile for those cells, which means that you need to have the boundaries of those cells. And so we've been developing methods that allow us to define those boundaries. One of the ways we have done this is to couple immunofluorescence with MERFISH. And there we will do immunofluorescence against pan-cell type abundant cell surface markers, such as the sodium potassium ATPase that maintains the potential in essentially all cell lineages. We can label our antibodies with short DNA oligos that contain readout sequences, and that now allows us to use the exact same repetitive staining process that we've optimized for MERFISH to read out both the identity of RNAs as well as the location of these antibodies. Now, there's a second approach that we have also started to leverage, and this is in collaboration with Peter Karchenko's lab. Peter has a bioarchive paper highlighting an algorithm known as Baser, and we've had great luck using Baser to help us define the boundaries of cells. The notion here is that individual cells have their own unique transcriptional profiles if they are distinct cells. And so by looking at the local neighborhood of RNAs, you can make estimates of what RNA should be grouped together within individual cells versus different. And that's the central idea that underlies Baser. And what I want you to see here is that we're able to use this approach with no prior information to define the boundaries of transcriptionally distinct cell types. We can then couple in the immunofluorescence to provide prior information to further refine this type of information. And with that, we can define the cell type diversity we expect to see in the gut. What you see here is a UMAP representation. Again, individual cells are markers colored via a Leiden clustering approach now. And we can see the rich diversity of cell types you expect to see in the gut, marked by the markers that we would expect to see and with the spatial distributions that we would expect to see. And so this is work that is just getting geared up now in my laboratory. And I share this with you just to emphasize that this is a technology that we are now working to extend to many more tissues than just the brain. And we can get the same type of rich insights from the cellular atlases that we are creating in these tissues. So with that, I just want to leave you with a summary. What is MERFISH? MERFISH in many ways is a series of single molecule fish images, all performed on the same sample. But in each image, we target a different subset of RNAs, such that across this stack of images, each unique molecule has its own fluorescence on-off pattern, which represents a binary barcode that allows us to identify that. And what's exciting about this is we've shown that this idea can be leveraged to massively multiplex single molecule fish. We can identify hundreds to thousands to tens of thousands of different RNAs. And by leveraging these error-robust and correcting encoding schemes, we can actually do so with very high performance, near 100% detection efficiency, very low detection limit, very large dynamic range. And one of the points that I have highlighted only in passing is that this can be a very high throughput technology. We can quantify tens of millions of RNA copies in tens of thousands of cells per day. And this type of throughput is only scratching the surface of what is actually possible with existing technologies today. And so what is MERFISH? It is a tool for genomic scale microscopy that allows us to create nanometer scale maps of transcriptome scale expression in cells in their native context, in cell culture, in tissue slices. And with that ability, give us the ability to not just define the parts of these systems, but to tell us how those parts fit together. So with that, I just want to acknowledge the tremendously talented group of people I have had the privilege of working with, both as a postdoc and in the new lab that I have recently started. So I want to thank the members of my postdoctoral mentor, Xiaowei Shuang's laboratory, who were involved in the development of MERFISH with myself. So Xiaowei was a monumental player in this effort and has just helped, has driven the development of this technology. It was a tremendous experience to work in her laboratory. The tissue work that I showed you in the hypothalamus was done in close collaboration with Catherine Duloc's lab. It was a pleasure working with them. I want to highlight Stephen, DJ, and Eric, who were involved in that tissue work, and how Alistair, Stephen, Junji, Guoping, Tian, Hazen, and George, who were all involved in different aspects of developing and extending the MERFISH technology. I also want to thank the new members of my laboratory. I have just been so unbelievably fortunate to have a talented group of people to start my laboratory with that you see here. And I want to highlight particularly two people, Paolo Codno and Raj Lanshu, who are the team that are driving some of our efforts in the gut with MERFISH. And I gave you just a highlight of some of the work that they're doing now. I also want to thank our collaborators in Peter Karchenko's lab and Roni Nawarsky's lab that have been involved in aspects of the work that I shared with you here today. So with that, again, thank you so much for this opportunity to present my work with you. And I'm happy to take any questions you might have.
So thanks, Jeff. This is absolutely fascinating and stunning. Super cool technology. I do have a few questions, but it seems like already some questions in the chat box. Are you able to see the questions, or I can read for you?
I think I can pull them up. Yes. Would people like to unmute themselves and ask their questions? Otherwise, I can read the question and then give an answer.
Yeah, feel free to unmute and ask. I kind of like that more interactive format. If you are able to do.
Well, if there's note takers on that, I can go ahead and read some of the questions. So one question is, could you comment on the balance between increasing tissue or section thickness to get more 3D info versus probe hybridization and washing step times, as well as microscopy limits with regard to resolution? Great questions. So we don't actually believe that probe penetration or washing are going to be particularly problematic as the tissue slices become thicker. I didn't have time to highlight this, but one of the technologies that we developed and has been integral to everything we're doing with MERFISH is a tissue clearing approach, wherein we embed our samples in a hydrogel and then remove everything. Those hydrogels are typically 50 to 100 microns thick already. So we have a 10 micron thick tissue slice that's been digested, sitting at the bottom of 90 additional microns of gel. And we can already get probes in minutes. So we don't envision that aspect is going to be problematic. The microscopy is perhaps the bigger challenge because we are doing very high numerical aperture imaging, and that typically requires oil immersion lenses, which are corrected really for the immediate vicinity of the cover slip. So the further you go into these samples, the more scattering, the more aberration, the dimmer the signal. And so that's why we typically image somewhere on the order of 10 to 20 micron thick slices. We have another question. Okay, actually it's the same question. How do you see extending the sensitivity of MERFISH from 2D to 3D? It's a great question. I think that moving into 3 dimensions is one of the next directions for spatial transcriptomics technologies, and image-based approaches, I think, are a good avenue towards doing that. There are many ways that this could be done, whether it's through serial reconstruction, which is the type of thing that we can do right now, or more advanced microscopy techniques that involve things that correct these aberrations that increase with density, like adaptive optics, or potentially light sheet microscopy. And I had a question about talking about sample preparation for MERFISH. So yeah, honestly, there are maybe 2 things to keep in mind in terms of sample preparation for a technology like this. The most important in our experience is the quality of the RNA. And so you really need to be able to prepare good sections that are prepared in a way that preserve RNA integrity. If you can perform single molecule FISH on those sections, then in our experience, that's good enough to go straight to MERFISH. Outside of that, there are only a few things that we do different in sample prep with MERFISH. So it's largely a single molecule FISH preparation with the addition of this hydrogel embedding and clearing, which improves the background and non-specific binding. Honestly, whenever we do single molecule FISH, we also do that hydrogel clearing because it improves the background in that context as well. And so that's the set of questions in the chat.
I think Don Fernandez raised the hand. You can go ahead and unmute yourself and ask.
Hi, I just had a question regarding how reliable it is to compare samples from one to the next. So let's say you have, 10 cancers from 10 different patients. So I know at this point there's a lot of batch correction algorithms for single cell RNA sequencing, but how does that relate to the spatial transcriptomics? Is it possible, what's happening for the future?
It's a very good question, Don. I think it's important to maybe divide this into two possible confounding factors. The first is, of course, your measurement and the sample prep that went into that. Honestly, we find in the mouse hypothalamus, which is a very reproducible tissue, that we don't really have problems correcting for some modest variation in measurement performance. Typically, that shows up as a slight variation in our detection efficiency. That we really can just normalize our detection efficiency across samples, and samples will co-cluster. The second confounding variable is essentially the degree of spatial reproducibility in your sample. So if you have a sample that is very stereotyped and you can come back and image the same thing in multiple samples, then I think that this performs very well. If you have a highly heterogeneous sample, perhaps tumors are a good example of this, one or two slices from that tumor may not actually be very representative of the entire, the entire transcriptional and spatial patterning in that tumor. And so then you end up in this situation where it's not that the measurement was a problem, it's that you're undersampling the biology there. And then one measurement to the next is in some sense a random sampling of the much richer diversity that's present. That's, of course, going to vary sample type to sample type, but it underscores why I think we need to be pushing all these technologies, including single cell sequencing, towards millions and millions of cells. There's the ability to statistically power these measurements, I think, is going to require much larger data sets than what we're collecting today. And what's exciting about these image-based methods is that in principle, there's a clear roadmap to achieving tenfold increase in throughput.
Thanks so much.
Yeah, thanks, Jeff. Actually, that's no question we have been discussing within our community as well. So in single cell sequencing, so we're able to do batch correction of kind of some batch effect normalization, but how to do that for spatial? I think in my opinion is you may not have to. Instead, what you should do better is to really correlate your single cell spatial data with the tissue histology and with the imaging, and you really see the difference within each individual sample to kind of, in that case, you don't have to compare across different samples, but really kind of benchmark against the standard tissue pathology data. I think our pathology friends are looking at every day. But eventually you want to study a large cohort and what to do at that stage, then that's no longer batch correction, that's my opinion. But we do need to think about how to do data harmonization and how to statistically compare. I think kind of sampling, shallow sampling, that might be an issue, but that's the same thing when people do kind of needle biopsy, whether or not you are sampling the entire tumor, right? So I think a pathologist might give you some advice, and we need to work with them very closely to address this question.
Yeah, that's an excellent point, Rong. I really agree with everything you're saying, and it emphasizes that as you move to clinical samples, the limit may not be the measurement. The limit may be the amount of tissue you can actually get.
Mm-hmm. Yeah, I have a few questions, Jeff. I think once in your data, you've shown inhibitory versus excitatory neuron. If I remember correctly, you don't see a lot of excitatory neuron, but in theory, you should have 4 times more excitatory neuron compared to the inhibitory neuron. So maybe that's specific region your image.
That's exactly what it is. The excitatory inhibitory balance is this is what you expect in the hypothalamus in the preoptic region. It's predominantly inhibitory and less so excitatory. So we really nicely recapitulate what people have already shown with inhibitory and excitatory markers in terms of both the abundance of those neuronal types as well as their distribution.
Mm-hmm. Okay, I see. I think I need to learn better neuroscience. So I think another question I have, or maybe more philosophical thinking, which you talk about the low copy number genes, and so I think a MERFISH and a SM fish, in theory, gives you 100% capture efficiency, right? That's huge benefit for sure compared to single cell sequencing. And but I think in biology, if only 2 or 3 copies expressed for that gene, I think most of the people don't believe that particular gene or eventually the protein you've transcribed gonna be really functioning. So I think biology is not really, and it's more like a digital. So you need to have enough transcripts expressed to kind of induce that type of function. So I think my thought is very likely just a few copies of that gene expressed indicates the cell is kind of poised to make some commitment towards a different fate or do certain function, although that cell has not started to do that. Maybe epigenetically already reprogrammed, but the expression of that gene isn't significant or even not expressed or expressed at a very low level, but the cell's gonna do that anyway, just a matter of time. That's what I think. But very unlikely 2 or 3 copies gonna make huge difference in terms of cellular function or fate decision.
I disagree. I think so. I think that that's probably true for some genes, and certainly we know that that the cell could tolerate some transcriptional noise in some classes. But there are definitely classes of genes where a couple copies of the RNA are all you need to give rise to the function, the protein that gives you the functionality you have. And I think we see this. So for example, in many of the receptors that we are actually showing, these are defining neuronal types for which we already know their responsiveness to those neuropeptides or hormones. So we already know that those neurons exhibit the functionality that one would anticipate if the receptors they expressed are actually giving them that functionality. And so we already know that that's the case in those handfuls of instances. And I think this also explains why, when you go through the single cell RNA sequencing data, there are certainly transcription factors that pop up, but as a whole, marker genes tend not to be enriched in transcription factors to the degree that I suspect we would anticipate they would be. And I think that, again, transcription factors are these examples where you may not need a lot of copies of that protein to give you that functionality. And so I think you're right that there are certainly some genes where the function you would have in that cell is derived by the sheer volume of the protein you make. There's so much of it, it's required for that function. But there are definitely categories of genes where just a few copies of that RNA is all it takes to give that functionality to that cell.
Well, is that kind of related to the faster turnover of the transcripts, although you can see only a few copies, but actually very fast kind of synthesis and degradation rate of that gene. So you see a few, but actually, for example, within the past couple of hours, that cells actually made a lot already transcribed, but just degraded.
Yeah, I think that I think you're absolutely right. And, this is a place where I just don't happen to know the numbers of the typical multiplicity in terms of protein number to RNA number. I suspect that actually could vary dramatically based on the lifetime of the RNA, the lifetime of the protein, and the translational efficiency. But I think, I think a couple copies of RNA almost certainly doesn't correspond to a couple copies of that protein in kind of a global average sense. And that may be one way to kind of fill this gap between your intuition that, if I had 3 copies of a receptor in a cell, is that really going to be functional? It's probably that you have many, many copies of that protein derived from relatively small, continuous, steady state store of that RNA.
Yeah.
I thought the multiplicative number was 20 or something. I remember Gordon Mills telling me that you got one mRNA, you're going to get 20 proteins out of it. But that'd be a fun number to look up. Maybe somebody else on the call knows that multiplication number, but I think it's something like 20.
Thanks, Justin.
Yeah, if you can do kind of time lapse MERFISH, that would be cool, and correlate it with the protein dynamics.
Yeah, it's a fixative technique, so destructive to that instance. But there are lots of cool things you can do if you have models where you can reproduce the system in the same state and do pulse chase style measurements.
Great. Maybe one more question from this side. For the gut and brain interface, right? The biology that you're studying there. So are these mostly the subset of cells talking to subset of cells, or are you looking at the transition of signal? What are your thoughts on this?
Yeah, it's a really, really great question, Ahmet. I mean, I think our perspective on this is that for many questions, we're at the atlasing stage. So this analogy I make of understanding the parts and how they fit together is where we see we are. We're not starting at all from scratch. So, decades of beautiful work have established through really like cell type by cell type, painstaking work, much of the diversity that's there. And single cell RNA sequencing in many ways has largely confirmed that work and added a few additions to it. But our goal really is understanding the diversity of cell types that are present and how they're organized and how that organization changes in response to perturbations, a wide variety of perturbations. So that is our entry point. But the idea is that once you can start to map these cell types, because many interactions are local, now that's a hypothesis generation tool for how these cells actually interact. So for example, in the brain, we're looking at peptides and receptors. So we can ask, what are the peptide receptor pairs that this neuronal type expresses? The galanin receptor, one of the galanin receptors, and this neuronal type expresses galanin. Now we have a hypothesis that these two could be part of a circuit. And so that's the type of thing then you then follow on with, any of the variety of tools to now functionally validate those things. So in some sense, these atlasing efforts, I think, give rise to these functional connectivity hypotheses, whether it's in neuronal circuits or it's in mucosal immunity. So.
Yeah, that's very exciting. Actually, I remember DS-Chem on the fly genetics had thousands of variants. And, some of the genes in the brain and gut interface actually might have a lot of variants that MERFISH can really pick up and explain that interface. That's a pretty cool, actually, platform to study. Should be exciting.
We're excited.
Yeah, we are very excited. I think one last question in the chat box, maybe just, that's very last question, and then Ahmet can conclude.
Oh, I see. Just added in now. So let me stop that. Lo, do you want to ask your question, or I can read and answer it?
I can ask it.
Great.
It's beautiful talk. Thank you. I've been analyzing some MERFISH data myself, and I was wondering how you translate those usual analysis tools that we use for 10x or droplet sequencing data to this data, because those tools often rely on a very large number of genes to extrapolate background levels or build a null hypothesis. And we don't have that many genes when we run a MERFISH experiment. And often we just want to condensate in there all genes that are relevant and will vary between conditions or between cell types. And there's no really background level for anything.
Yeah, this is an excellent question. I think there are a couple things that we do, but let me just start by saying, I think it's, to be provocative, I think it's the Wild West. These type of data, whether it's MERFISH or other image-based approaches, are really very new. And so I think that as we saw with single cell, hand in hand with the experimental developments that give us these data set, need to come computational developments. And this is something that we're working on. It's something that we're collaborating on. I'm just very excited, excuse me, with where the field is going on this. What we have been doing so far is leveraging established single cell analysis toolboxes with just a few tweaks representing aspects of what's happening in the MERFISH data. So the first is that we normalize not by total number of counts that we see, but rather volume. We have a volume for each of our cells. We normalize by volume. We find that works quite nicely. It also nicely addresses the fact that in slices, you may not image an intact cell. You might get the bottom third of a slice and the top 2/3 is out of your slice. So you got to address that issue. Volume normalization does a nice job of addressing that. We have a few really modest batch effect approaches where we recognize that some data sets, spots are a little brighter, you detect 5% more or something, we'll normalize those differences. After that, what we do is largely the standard analysis without typically dimensionality reduction. We have sometimes identified principal components and then run the rest on those, but often the principal component analysis is there to make it more computationally tractable. We find that as computation resources have increased and the more modest number of genes we have, though often we're looking at hundreds of genes, we don't need to make those concessions. The last thing that we have found is that, of course, there's a standard, excuse me, in the single cell field to do this logarithmic transform to help address this dynamic range issue. Nothing necessarily right or wrong about that. We often don't do that because MERFISH does have a false positive rate. It's very low, but that is real. And when you do this logarithmic transform, you can accentuate the impact of that false positive rate. It doesn't make a huge difference whether you do that or not, but often when we've clustered it, we haven't done that logarithmic transform. Outside of that, there aren't really any modifications we do to the standard tools.
Thank you.
Great. Joe
Got
has a
time for
question.
another one, Ahmet or Rong? Can I ask a question?
Sure, please.
Yeah, I love this technology, Jeffrey. So, Jeff, along that theme, we have like controls that are like what are called the ERCC, the alien sort of species control backgrounds, and then you have sort of I don't know what you call them, false code calls, where you're calling something that isn't even in there, and we typically collect both those numbers. Can you expand? How important do you? What's the role of those two controls in these types of experiments? Do you believe?
Yeah, so we don't have any dosed-in RNAs that we've ever done. So the analogous measurement of the ERCC controls does not exist for what we have done. It's interesting to talk about whether that could be done or not. Honestly, never thought about it.
We actually do it on all of our experiments. We find it very useful. We're working on this thing called a spatial molecular imager.
Yeah.
And also on our DSP, we always dope in ERCC controls. And we find them to be pretty useful control reagents. Pretty easy to do.
Yeah, I guess, the key is how you get them to stick. And with our hydrogel approaches, I think we could probably embed them in the hydrogel, right?
Oh, that could be. I hadn't thought of that. We're just doing normal in situ hybridization. So I hadn't thought about, you're right. That adds, I guess, a complicating factor to that.
Yeah, I mean, it's a very interesting control to do. For us, we always include these blank controls. These are barcodes that there's nothing there. And we see them, right? And that's our false positive rate. And what we like about that is that it's embedded in every measurement, as you know. So you got a false positive rate on every single sample, every time.
Yep.
And as we have developed the next generation of decoding, right? So you know, if you like, there is the base calling analogy here. How do you take a set of spots across a stack of images and decide what's where? And that's the analogous problem to base calling. We've got algorithms that we think are very high performant, but I don't think this is a solved problem at all. And I think that there's a lot of innovation that can happen here. Those false positive rates have started to play more of a role in how you define automated ways to divide foreground and background in these type of, if you like, decoding algorithms, or we call them, or the analogous to this base calling problem. And so, yeah, I think we always include them in there. The other thing that's nice about our error correction approaches is that we get a per bit error rate. We know exactly how many RNAs had to have error correction applied. We know exactly what bit, and we know how frequently in all of this. And so we can immediately see if there was a problem in round 7, those corresponding bits, we see an uptick in their error rates. And so, you know, those are the type of things that are embedded in these highly multiplex data that we typically use. The ERCC is an interesting control. We'd have to think a little bit about that.
I'm with you on the unsolved problem. I wonder whether we could think about doing a hackathon on something on how to, I mean, it seems like a hackathonable kind of thing, Jeff. I don't know, but
look, the more people involved in this, the better. I think this is the aspect of the exciting thing about this time. And what I would say is, at its core, there is a growing suite of these image-based transcriptomic techniques, as you know very well. And they are all binary barcodes. It doesn't matter what colors you call them. At the end of the day, every technology, even the in situ sequencing technologies that are reading out, you know, sequenceable barcodes, are a monochrome stack of images that you're on or off in. And so this is the thing that I would hope would incentivize the next generation of bioinformatic tools, in that, you know, if you can solve this once, you've actually solved it for a whole field, which of course is probably not quite true. There are enough differences that might make some distinction there, but there is a common informatic problem here. Yep. And I think that makes for an exciting opportunity. We've thought a lot about this. We're working on some things here, but, you know, I think this is a place where more cooks in the kitchen is better. You know, let's get more people involved in this.
Because, you know, all the colors come out, even those monochrome planes, they come out slightly off registered, and you got to bring them all together. Then you got to figure out which one's on a single RNA and which one's on just two closely spaced different RNAs. I think it's got a fertile room for a lot of interesting software to be developed for that.
And the thing to keep in mind is that, honestly, a lot of this is well-trod ground. So some of it is just reinventing the wheel. Part of what we do in terms of our decoding is we leverage concepts called soft decoding. So I don't know of anyone else that has really deployed this yet, but it works amazingly well in us. If you look in the history of the MERFISH development, our per bit error rates dropped by an order of magnitude, and it's because we adopted soft decoding. This is well established in the binary space. It just says that, look, you don't make a hard decision on whether something's a 1 or a 0. You just represent that it could be a continuum, and you leverage that so that information in other bits where you might be more certain helps you make the decision on this bit where you might be less certain. It's a brilliant idea, and it's played out really well for us. And it also turns out to be extraordinarily computationally efficient compared to some other approaches. But this is the other thing, is that, as you probably know, generating a terabyte of image data in a day is the easy part. Getting that terabyte of image data into analyzed something in a way that's not breaking the pocketbook in terms of cloud compute, that is another place where we could really see some innovation.
Yep, I agree completely. Super interesting things.
Yeah, I like a hackathon idea. To think about.
I do think we just got done with our first hackathon. I'm just surprised that the submissions were so good, and we're definitely going to do it again. And this is one area that I'm really thinking about. There's so much innovation left to be done here. It's really,
Completely agreed.
Totally. So any other questions, or should we leave that for the emails? What would you prefer, Jeff? Continued questions, or?
If anyone has any other question they'd like to ask, I'm happy to stay around for a little bit more. And of course, if anyone would love to follow up by email, I'm always happy to answer questions. Looks like we have another question now.
Yeah.
Hi, Jason. So maybe just one quick question. So you mentioned about false positive rates. So what's actually the number there? So is it like 0.1%, 1%? So what's the typical?
Yeah, typically what we
range
find,
there?
you know, as a rough rule of thumb, it's like 1 false positive per 10 to 30 cells. It depends on the library. It depends on the tissue. But that's the rough range for what we see.
Thanks.
And there's also a question on the chat, and Gabriel is asking.
Jeb, do you want to ask your question?
Yeah, just high level. How is MERFISH and SeqFish? Are they different? It seems like they're both great techniques, but you know, high level seems like it's kind of a similar core.
Yeah, I mean, I think we have you know, great people on the call here to answer this. You know, MERFISH and SeqFish were developed independently, essentially at the same time. The original MERFISH implementation looks very similar to what we have today. The idea was that we had a 2-step labeling process where we create these encoding probes, they go on, we have these readout sequences, the readout sequences are the embodiment of the barcode. That has all sorts of substantial advantages in terms of the throughput of the technology. The binary idea with error correction was built in right from the beginning. And there was a recognition that this ability to set the Hamming weight of the barcodes gave us a lot of flexibility for applications in the future. And it was a monochrome measurement. And you know, SeqFish in its first implementation was direct labeling of RNAs, followed by removal of those probes by this very clever enzymatic approach, and was very much a colorimetric barcode, which built on this beautiful history of colorimetric barcoding from, you know, really dating back in the fish community to Rob Singer, and recognizing that this colorimetric barcoding could be increased by adding rounds. And so, you know, over the years, a lot of what we have implemented has been incorporated into the SeqFish protocols, which is really great to see. And so I think at this stage, you know, the distinctions are modest between the technologies.
Cool. Thanks a lot.
Are there any other questions? If not, let's thank the speaker again, and thank you. I'm going to use this clapping sign. And and before we wrap up, I'd like to also announce that next week we will have Dr. Dennis Wirtz and also his colleague Ashley Keeman from Johns Hopkins University, and they'll be talking about 3D pathology using CODA. And with this, I think we're happy to wrap up today. And please enjoy your long weekend holiday, and hope to see you guys next week. Bye-bye.
Thank you. Thank you very much.
You guys.
Yeah.